So here's my speaking notes, and this is the list of everything that I'm going to be sharing with you. So I so you can take a picture of this, but I also made a QR code for it. So if you scan this QR code, then you can get to the Google Doc that has a list of all the different apps and programs that I'll be sharing tonight. And first, I'm going to share all of the different Android apps that I use. Well, first, I'll introduce myself. My name is Alan Rockefeller, and I've been studying mushrooms since about 2001. And I first got into mushrooms when it was Christmas Day 2001, which is my birthday, and I was hiking in the woods, and there was mushrooms everywhere because that's the peak of the mushroom season where I live in California. And so I decided that I wanted to be one of those people that could identify all the mushrooms and knew what all of them were. And so I started paying attention to them. And the more I paid attention to them, the more interesting they got. And at the time, I was working for the government, breaking into computers for NASA. And then around 2010, I um, I decided that mushrooms are more interesting than computers. And so I decided to quit my job and just be a mycologist full time. And I've been doing that ever since. So I still like computers and I like to mix computer stuff with nature. And I think it's like a really good kind of counterbalance. So I can spend like half the day out in nature and the other half of the day in the laboratory doing PCR and DNA barcoding and microscopy or using my computer skills to study the mushrooms. And so I'm going to show you all the different apps that I use on my phone and some of the computer programs that I use to study mushrooms. And so we will connect my phone up here so you can just see what's going on my phone. All right, I think that worked. Do I have a bar on top of it? I should move this around. Can't really.
There's a little.
Is there a little hider under more?
Yeah, like hide
Yeah.
floating moving controls. Yeah.
Awesome, thanks. All right, so the most important one is iNaturalist. And the way I use iNaturalist is that whenever I find a mushroom, I hit the plus to make a new observation, and then I click No Media. And now you can see it's finding the location, and the location accuracy gets more and more accurate as it starts to get more and more satellites. So who knows how many satellites it'll get in here, but it stops trying once the accuracy is 10 meters or less. So usually I'll wait for this number to go under 10. Um, and then I just take a bunch of pictures with the regular camera app on my phone, and then hit the plus over here, choose image, and just start adding all of the pictures out of here. And that way I can just like snap a whole lot of pictures really quick. And I don't have to, use the slower method of taking them one at a time through the app. And then it always gets the GPS coordinates really good. And then I go over to the collection notes, and in here, I just add all of the notes, all of the things that are not obvious from the photos. So that's the taste, the odor, the smell, the texture, nearby trees, all that kind of stuff. And I can hit the microphone, so I can just tell my phone that instead of having to type all that in. Another really cool thing about iNaturalist is the Explore feature. So if you go to Explore, then it shows you all the nearby observations. And so you can search like for chanterelles or whatever you want to look for. And it'll show you where all the nearest ones are. Or you can like make it only show your observations so you can see where you've been, all that kind of thing. So I use this a lot. I put every single mushroom I see on there. And if I see any plants, and I don't know which plant it is, I put the plants on there. Birds, pretty much everything I see in nature. Does a really good job of keeping track of everything. And I do a lot of camping. And so one thing that I have to do when I'm camping is to get cell phone service so I can upload all of my photos. And so I have a bunch of apps that help me get good cell phone service. The one I use the most is Network Cell Info Lite. And this is a free app. All these apps will be free, and I'll mention if they're not. But it talks directly to the radio in your phone, the cellular radio, and shows you the exact signal strength. So you'll notice, like if you're in some kind of like Airbnb, like 10 miles away from the tower, you might put the phone in one window and it'll 1 megabit, and then you put it in another window and your internet will be 100 times faster. And so being able to see the exact signal strength, you just move around the phone until that gauge goes up as far as you can, and then you can get the fastest cell phone signal. You can also go over here and go to map. And then as you like walk around to drive up and down the street, it will graph the signal strength of the cellular tower right on the map here. And so you can see like where is the strongest signal to set up your tent and set up your campsite so you can upload your photos. Another really cool is Cell Mapper. And this one shows you all this cool information, like the frequencies that you're on and all of that. But you can also go to the map and you can see where the cell phone towers are. And so here's kind of a map of the area. We'll make this area. So you can see Denver has really good cellular coverage, but these red areas are like dead spots in the city. And then you can see where all the towers are. So let's see if we can. I don't even know if I can even find myself on this map because there's so many people, but let me just zoom out and go over to like National Forest area. You can see there's not nearly as many towers, but say you're looking for a campsite, maybe you would zoom in over here. And this looks like a pretty good place to camp because there's a tower out here, and then I can click on the tower and comes up with some data, but it shows you which direction the antennas are pointed. So like most rural towers, they point antennas just right up and down the highway. But now you know there's some areas that you can maybe set up a tent there and get really fast cellular service, like maybe right in here would be good, like the end of that road there, something like that. So it's really nice just to be able to see exactly where all the cell phone towers are. And then when you're out there and you want to upload your photos, I just go over here and turn on the mobile hotspot and then connect to that uh, through my computer. But usually the cell phone company will throttle mobile hotspots. Like I have visible service, which is like Verizon. It uses Verizon towers, but it's only 25 bucks a month. And it's great service, but they throttle your mobile hotspot to 5 megabits. But your phone is not throttled to 5 megabits, so the cell phone company tries to detect if you're using your computer or using your actual phone. And so if you set the packet time to live on your computer, then the phone company will think that your computer is your phone because it has the same time to live on the packets. And I will show you how to do that. So here is PowerShell. And if we just ping localhost, you can see the time to live. That's right here. That's the packet time to live. And the packet time to live is 65. And so what happens is every time it goes through an internet router, that time to live goes down by 1. So when it hits my phone, it goes to 64. And the time to live on my phone is 64. So then the packets are the same coming out of my computer or my phone. So instead of being throttled to 5 megabit, I can get whatever speed is on my phone, often hundreds of megabits. So you can upload your photos really fast. And I can see my packet time to live with this command here. So you can see it's set to 65. Default on Windows is 128. And then if I want to change it, you go just like this, and that's the command to change your packet time to live. And I haven't found any negative impacts from doing this, so I just leave this computer always set to 65 packet time to live, and that way it works really fast when hotspotting through a phone. Also, if you guys have any questions, feel free just to shout them out. It's totally fine to interrupt me, but I can also try to take questions at the end. So another one of these cellular programs is OpenSignal. And um, This is another one where it shows you a map and you can see like the speeds. So you can see what, like how good the cellular coverage is, and it's kind of crowdsourced all that data. And then it can also show you directly where the tower is. So this is now pointing towards the cell phone tower. And then one I just started using recently is called NPerf. And this one, yep. Oh, we're still over here. Thank you. Okay, NPerf. Um, let's see. So really quick, open signal. Here it's showing me the direction of the cell phone tower. And here's the map on open signal. So you can see where there's good signal. But NPerf, it's a little bit more interactive because what you do is you start active mapping and then it just like leaves a trail on the map um, showing how strong the signal is. And then it makes that data public and it adds it to the permanent public maps. So if you're like in the middle of nowhere, you can just like turn this on as you're driving around and then you can see where all the good signal is. And when you come back later, that data will still be there and anybody else can use the data and see where the good signal is as well. Another one that I like is Sun Locator. And what this does, I use it for a few things. One of them doing bioluminescent, looking for bioluminescent mushrooms, it has to be super dark. So this will tell me when. And they added all these annoying ads and they make it really hard to cancel. And that's an annoying thing. They try to make you pay for the app by putting in the really annoying ads. So I think you get the idea. I'm scared to open that app again. But it tells me like when the full moon or the new moon is, and then if there is a lot of light coming from the moon, if you're looking for glowing mushrooms, then it's going to be like everything in the forest glows if there's any moonlight. So I will see when the moon rises, when the moon sets. So either go out in the evening before the moon rises, or get up at like 3 in the morning after the moon sets, and you can have really dark skies and find all the glowing mushrooms. But the other really cool thing about this is that it shows me the exactly where the sun is. And it's really tempting to open this app again because it's really cool, but I'm scared. But basically you open up, it opens up the camera and it shows you exactly where the sun is, and you can see exactly where the sun will and at what time it'll be at each point in the sky. So if you're like camping out in the desert and there's like a big boulder there, you can be like, okay, if I put my tent on this side of the boulder, the sun will hit my face at 10:42 in the morning. And just like clockwork in the morning, like 10:41, you'll notice like lots of sun. So it's really good for just finding exactly where to put your tent to get the shadow in the morning. Another one that I like is Waze. And Waze is very similar to Google Apps, but has Google Maps, but it has one feature that Google Maps doesn't have, and that's that it has a speed alert. So let's see if I can go in here. I think it was settings and alerts and speedometer. So here I have, it shows the speedometer, but if I'm speeding more than 15 miles above the speed limit, then it dings my phone and plays it over my stereo. So if I'm just driving along and I don't notice that all of a sudden there's a 25-mile-an-hour zone, then my phone will start beeping at me and I will notice before the police do. Another thing that I like to do is a lot of times, if I'm like using iNaturalist to identify mushrooms, and let's see, let me just go into my observations here. And if I'm just like, yep, oh, we got Um, let's see, well, actually, the important part, well, I'll switch back over to this, but the important part is just that with iNaturalist, if we like try to identify something, oops, try to identify something here. So it'll try to identify this, and it's giving me a few different options. So I know this plant is a Thalictrium, but I don't know the difference between Thalictrium fendleri, Thalictrium sparsiflorum, Thalictrium occidentale. So what I will do is I will go over to ChatGPT and ask it what the differences are between those things. So if I go over here, like what are the physical differences between Hyphiloma fasciculare and Hyphiloma capnoides? And it gives me kind of a nice breakdown of what the differences are, so I don't just have to stare at the photos. Another thing that you can use ChatGPT for is to construct iNaturalist URLs. So there's a lot of really powerful things you can do with iNaturalist URLs, but it's not too intuitive to be able to build these things. And so here, I just ask it to make a URL to show me all of the all of the DNA barcoded things in Colorado. Let's see. And so if I just copy this, going to new tab, and here it is showing me all of the DNA barcoded observations in Kentucky, even though I ask it to do Colorado. So that's an example of a hallucination. And ChatGPT will very confidently lie to you. So then you can just change this to Colorado and see that we have 1,062 sequenced observations with photos on iNaturalist from Colorado. And this is a really useful thing for when I'm like trying to identify mushrooms in Colorado. For example, say there is a rusula, and I want to know which rusulas really occur in Colorado and not just which rusulas people misidentify in Colorado. You hit this, and now we have these are 19 species of rusula that have been found in Colorado and have been identified with DNA barcoding. Another app that I like to use are these mapping apps to track me. And so one I use a lot is called Strava. And this is a free app. And it's supposed to be sort of like a social media for exercise, but I just use it to like track myself. So if I just start this when I park my car, then it draws a map with this line behind me. And if I like set down a knife or a basket, I can track myself exactly the way it came back. Google Maps will lead you back to your car, but if there's like maybe a cliff or something like that, and there's only one way back, you won't really know exactly how you got there. You can also kind of do these things. If you just click on something here and then click on this, you can like see this kind of like, and here's one of my hikes in Telluride. And so this is when I was scouting for the night hike. So I led some night hikes there and I kind of checked out these places. I went to Bear Creek and I saw 4 different bears on that little section right there. So there was tons of bears in Telluride, but it's kind of cool how you can kind of see like in this 3D view of exactly where you walked. So that one was Strava. There's also Avenza, and Avenza is another mapping app, but on this one, what you can do is load in custom JPEGs and then see where you are in the JPEG. So a lot of like biologists will use it. So they have your study plots all loaded in here and you can see where you are on the study plots. Or you could like take a photo of the area with a drone and then load that in here and see exactly where you are on this like high resolution photo that you took. Another one that I like a lot is All Trails. And so with All Trails, just shows you where all the trails are. And this is one that has a paid version, but the free version is just fine because I don't need to download this stuff off of, for offline use, but it's just showing me the nearest trails. And you can like click on them and see reviews and what people are saying about them. So that is a pretty nice thing when you're just like in an unfamiliar area and you want to know where the nearest hiking trails are. Another cool kind of map is called the Open Street Map. So OSM Viewer displays OpenStreetMap. And OpenStreetMap is a little bit like Google Map, except that it shows you a lot, it shows you very different things, and it does a much better job with like boundaries and things. So here you can see exactly where the boundaries are for the different national forests and stuff like that. That one is OSM Viewer. There's a couple more that are a little kind of similar, like CalTopo, another one that shows you topo maps, or the one that James Shaleen uses is US Topo. And with that one, you're loading in the USGS topo maps, and then you can like choose all the different USGS maps and see where you are exactly on the map. Another one we use that's sort of expensive is Onyx. And the really cool thing that Onyx does is that it shows you exactly who owns all the property. And so if we just maybe zoom in around here, and I got to go to the layers and turn on private land. And now you can see exactly who owns each house here, at least who the registered owner is. And so you can, some people use this like if they want to hunt somewhere, they know who to contact to get permission to go hunt on their land. Or you can just see where the boundaries are so you don't accidentally wander onto private property. This one's like $60 a year, but there's this other snowmobiling app called Polaris, and this one is free. And it does the same thing in that you can zoom in and it shows all of the property ownership. So a lot of people, I guess snowmobilers like to know whose land it is. And so this free app will show you the exact same stuff that Onyx does. And that's out of the property records databases. Another one that I use is SnapBridge, and this is Nikon's camera app. So SnapBridge does a couple things. One thing it can do is connect to your camera, and then a lot of Nikon cameras don't have GPS. And so this will use the GPS from your phone, and whenever you take a picture, it will encode the GPS as found by your phone into the picture. So you can have exact GPS coordinates on all your photos. The other thing you can do is have it automatically download all of the pictures that you take instantly onto your phone. So if you take a good picture with your camera, comes onto your phone instantly, you can create an iNaturalist observation and put the good camera photos on there instantly, right there when you're in the field, and not have to go back and add it on your computer later that evening. Yep. Yes, as long as you have it turned on. So I think most of them have it turned on by default. But if you're on an iPhone, you have to go into the system preferences and then privacy and then location and then camera and make sure it's turned on there. And if you're on Android, it's usually directly in the camera app. So if we were to like open up the camera app and go into the settings, let's see, where are the settings here? With that one. I don't see the settings on here, but if you do go into the settings, then there's like a add geolocation to your photos. And if that's on, then it does. And that's really nice because then when you add your photos to iNaturalist, it picks up the location. And so they're all in just the right spot. And you don't have to like tell it where you found the mushroom or anything. So that was SnapBridge. If you have an Olympus, then OiShare is the Olympus version of that. And then another cool GPS app that I use is GPS status. And GPS status, all it really does is it reads all the sensors on your phone. So here you can see where in the sky all of those satellites are. And then it reads a whole bunch of different sensors, but the one that I really like is the altitude. So it just shows me my exact altitude. And something like Google Maps will not show you your altitude. So Whenever I ever want to know exactly what else I'm at, then I just open up GPS status. And then there's a whole bunch of these other camera apps, and the main thing that I use them for is to do manual focus. So this particular phone is a Samsung, so I can do manual focus directly through the camera app, but most phones don't have any manual focus. And so if I open up open camera, or a few other of these camera apps, then I can set the manual focus on a whole bunch of things. One thing that this app does is that it can do photo stacking. So it can take a whole bunch of pictures at different focus depths, and then you can load those in and photo stack them with an app like Helicon Focus. I also like AccuBattery. And AccuBattery kind of measures your battery here. And so right now, how much power I'm using. But if I plug in a charging cable, then it tells me exactly how fast my phone is charging. And a lot of times you're driving from spot to spot. You really want your phone to be charging pretty quickly when you're going from one place to another. And so you can try with different cables and different chargers. There's a huge difference in the rate that your phone charges. And it also varies a lot by temperature. So if your phone's more than 100 degrees, it's going to be charging super slow. And then you know that you can just stick your phone into the air conditioning vent in your car and it'll charge like 10 or 50 times faster. And it's also a really good idea to carry a few spare USB batteries when you're out in the field. A cool one for seeing how much rain there is precip. And so precip has this really nice interface. And so we'll just go over here. This shows, let's see, we're in the past 7 days. So you can do like 48 hours or 24 hours. So this is just our rain today, rain in the last week. And it's a real nice interface, just like pan around and see exactly where the rain's been hitting. And see a whole bunch right out in these cornfields over here. Or you can do month to date. Or they also have a layer for soil moisture. And so this is kind of showing you how moist the soil probably is. This app is $20 a year, so it's not free, but I like the interface and I use it a lot, so I bought that one. But if I'm not going to do that, let's see, I think there's some free ones you can use. This is like water.noaa.gov, and it's kind of annoying to get it working right, because right now it's just showing me the rain gauges. But you can get this off of weather.com and a few other places. I had one of my bookmarks that was pretty good that Stephen Russell posted recently. But you can get that off of the Water NOAA website as well. Another kind of cool one is Weigel, and Weigel is sort of a hacker tool that will display all of the radio signals that your phone is receiving. And so it displays, here it's displaying all of the Bluetooth devices. And so this is all of the Bluetooth phones and devices from everybody in the audience, and also all of the Wi-Fi things. And it's kind of cool because if you're in the middle of nowhere, it'll log all of that. And like if somebody passes you on the trail, you can just look at this and see like what kind of phones they have. Or if you're in the middle of nowhere, you'll notice like a Wi-Fi network pop up all of a sudden, and that'll be like some scientific gear that's like up in a tree somewhere or something. But a more practical use for this is if somebody loses their cell phone, but their phone has Bluetooth on, then you can walk around and pick up the Bluetooth on their phone and kind of track down their phone, even if you can't find, when you're just like 100 feet away from it. It helps if you remember like what your MAC address is here, so then you know which phone is the lost phone. But just like in the middle of nowhere, if you lose a phone, just picking up any Bluetooth, you'd be like, yeah, I'm probably pretty near my lost phone. And then I also like Merlin. And Merlin will just listen to your microphone and show you exactly which birds are around. And it works really well. You can't really fake it out. At least I've not been able to fake it out by like whistling into it. He does not think that's a bird. But like when I was in Costa Rica a couple weeks ago, I hear some strange bird call, and you just open this up and it'll show you, it'll tell you exactly what kind of bird it is. You can download bird packs for the United States or South America or Asia or really anywhere you go. So that is a pretty cool app if you like birds. And then if you like stars, the really good star tracker app is Sky Portal. And Sky Portal is made by Celestron. And so, you just like aim it around and it shows you all the different planets and stars and just everything going on in the sky. There's also one called Watch, and Watch Duty will alert you if there's fires in your area. So you can have it so like if you're camping out and there's a fire starts near you, it can like alert you, or you can just like see where all the active fires are. So this is an app that's used by a whole lot of first responders. So that is all of the cell phone apps I have. So I'll show you some computer stuff. One kind of cool computer thing that not many people know about is Notebook LM. And Notebook LM is this AI tool that Google developed. And it does a few different things, but um, what you do is you load a document into it, and then it trains the AI just on this document. So for example, like ChatGPT is trained on the whole internet, but when you train it on only one document and nothing else, then you pretty much get rid of the hallucination problem, and then you can query this with questions about your document. And the way that I usually use it is I will load in like a big monograph, like a monograph of Mycena, monograph on Gymnopilus, and then I can ask it a question like, what are the microscopic differences between Gymnopilus and Gymnopilus subspectabilis? And if I wanted to find that information normally, I'd have to look at those two microscopic descriptions and then very carefully compare them to see where the differences are. And this will just tell you what the differences are. And then also it does this audio overview thing. And so an audio overview is what it does is it generates a podcast where it discusses this. So what I use it for is if you have some scientific paper and it's like really dense reading material and you don't want to read it, or if you want to listen to it in the car, it won't just read the paper to you, but it'll make this podcast where they have these two artificial hosts who are discussing it. And it does a really good job. It's kind of scary. It's like the first few times you listen to it, you're like, oh my gosh, I can't believe these are not real people. Because like they like make little jokes and they like, it doesn't just go based on the paper. It kind of like does a bunch of additional research, but it does a really good job of figuring out what the really interesting points of any paper is. And then, they'll discuss it and like talk about why the findings in this paper are really different from other papers. And it's a really good way just to be able to turn any dense paper into a very listenable podcast. It's also nice to be able to track things. And I have an Android, and unfortunately, all of the trackers on Android kind of suck. But let's see, looks like my phone's not connected anymore, but I use tile trackers, and they're very similar to iTags, but they're not nearly as good. So if you have an iPhone, then you're lucky because you can use iTags and they're really good trackers, but it's good to put one in your mushroom basket and you can make it play a noise, or you can just like, it'll show you where you left your mushroom basket, put it on your car keys, put it in your camera bag, all that kind of stuff. I have one in my microscope case. And that's nice. Just when I lose the thing, it'll just tell me where the last time I saw it was. And another program, this is one that I wrote. And what it does is it makes iNaturalist labels. So you go to images.mushroomobserver.org/labels. And then let's see, I think I have one open here. Yeah, over here. And so what you do is you put in different iNaturalist observation numbers. So we'll just like take this one. And so you can just type in the observation number or paste in a URL, and then it looks it up. So this one I just put in is a delphinium. So if you're making a bunch of labels for all the mushrooms that you found, fungi should come up in pink. If it comes up in blue, it's an animal. That one's a bear. If it comes up in green, then it's a plant. And then it shows you the username. So if you mistype a number, then you will immediately recognize that that's not your observation and and that you have the wrong number. And that way, what you do then is you go down and you hit print labels, and it will use the iNaturalist API to look up a whole bunch of things about those observations. And it will generate some very nice labels. And it gives you an RTF file. And that file can be opened up with Microsoft Word, or here I'm using LibreOffice, the free version. The labels look pretty good. They got the scientific name, the common name. You have the location, the coordinates, the location accuracy. And if you have any like DNA barcode information or voucher numbers attached to that observation, it'll print that on the label too. Also, um, any collection notes, like in this bear, I said the bear was eating berries, and that turns up there. And then it makes a QR code, and you scan that QR code, and it will go directly to the iNaturalist observation. And that makes it really quick if you're mailing in lots of samples for DNA barcoding, because the person who receives those samples and has to put a little bit of mushroom in each tube, they also have to type in that 9-digit iNaturalist number. But with these QR codes, they can just have a little $20 barcode scanner attached to their USB port, and then they can just scan all of your collections. And it's just like typing with a keyboard, except that you cannot mistype anything. And so when I print these, I like to print them on cotton cardstock, because regular typing paper only lasts about 100 years. But your herbarium labels will be, your herbarium collections will be stored forever. So you want these labels to still look really good in 500 years. So using cotton paper will make them last a very long time. And then I do a whole bunch of stuff with photos. And so what I do with the pictures is I connect my camera to the computer at the end of the day. And I don't like take the card out of the camera. I just plug the computer right into the camera, right into the computer. Because if you take the card out, then eventually you'll be out in the field and you'll have no card in your camera. And so if you just plug it directly in and load them that way, then you won't have to worry about remembering to put your card back in to the into the camera. So when you have all of the after I've loaded all my photos into Lightroom, usually I use a technique called photo stacking to take the pictures. And what happens is I tell the camera, like, take 50 pictures, and it takes a picture and then it changes the focus, and takes another picture and changes the focus. And you can see as I scroll through them here, each picture is a little slice of the final image. And so what I need to do is find what the last part of the subject is. And that looks like it is, let's see, let's try to move this bar out of here. So looks like the last image here is number 270. And then I'll scroll back through these and find just the first one. I'll just go back out of here. And so we will find the first one, and that's this one here. So this is the one that's focused, is really close. And then I will go down to 270, hold on Shift, select all of those, and then go over to export, Helicon Focus. And so now it's taking these 18 images and it's exporting them to Helicon Focus. And Helicon Focus is some pretty cool software, and it'll take these 18 images and combine them into one image where everything is in focus. And so that'll come up in just a second. But what this focus stacking does is it really bends the laws of physics. Let's see, I'll just hide that. Okay. Bends the laws of physics so you can get as much depth of field as you want, and it allows me to shoot with the aperture wide open. So then after the mushroom, it gets very blurry very quickly. So the subject stands out really well from the background. So I just hit render here, and it combines those 18 images into one image. And the resolution on this is really good. If I go over here and zoom in, you can see that it is razor sharp, even if you zoom way in. And so this is how I take almost all of my photos. If I don't really care about it very much, I will just use my cell phone. But if I actually care about the mushroom enough to want to get a good photo of it, I'll use the photo stacking. And you can see that the background gets very blurry here, so it makes the mushroom really stand out from the background. And after I'm done with run it through Helicon, I will save this and then load it into Photoshop. And there's a million things you can do in Photoshop, but typically what I will do is just straighten them out, adjust levels and adjust the colors so it looks on my screen just like it looked in the field. But there's also some cool advanced things you can do with Photoshop to like blur the background and all that kind of stuff. And sometimes, if I'm not on a tripod, I'm shooting with get really high, or shoot at really high ISO. So there's a whole lot of noise. And there's a bunch of programs you can use to reduce the noise, but we'll do Topaz Denoise AI. So with this program, this is made by a company called Topaz, and you can load photos into this program. And even if they're very noisy photos shot at a very high ISO, it will still take the noise out really well. And I'll just try maybe with this Mycomyces photo here. So this is a picture that I took at Telluride. And I use the base ISO of the camera, so it doesn't have very much noise, but this is an Olympus camera, so it does have some noise, and you zoom way in, you can see that it looks a little bit pixelated here. So the left is the original, and then the right is the preview, and you can see that the pixelation is gone on the right. So it does a really good job of smoothing this out, removing the pixelation while it preserves all of the detail.
Question.
Yep. Yeah, that's a really good question. How do we take the all of the pictures with slightly different depth of field? And so if you're using a phone, you can use something like Open Camera and just tell it to do it. But usually you want to do this with a camera and a macro lens. And the smaller the mushroom is, the more pictures you need. So if it's a really tiny group of Mycenas, I might take 150 pictures. If it's a really big Cortinarius, I might only need like 3 pictures. But there's 3 main ways to do it. The easiest way requires an expensive camera, but the newer cameras have what's called focus bracketing. And so I usually shoot with an Olympus OM1 or a Nikon Z7. And so Nikon calls it focus shift shooting. And so I just, on the camera, grab the focus ring and focus it until it's focused just a little bit too close. So the whole mushroom is blurry because it's focused too close. And then I go into the photo shooting menu, go to focus shift shooting. I usually tell it to take 35 pictures and hit go. And it'll take 35 pictures, changing the focus just a little bit. And then when it's finished, the mushrooms should all be blurry because it's focused a little bit too far. And if they're not, I could just hit it again. It'll restart from where it comes out. So Nikon calls it focus shift shooting. Canon calls it focus bracketing. Pretty much any high-end camera will have this. But some manufacturers have had it a lot longer than others. For example, Sony just got it. So with Sony, you have to have like a really expensive Sony body to use it. But Olympus has had this for like 10 or 15 years. So even a very cheap old Olympus can do it. So that's one way. Another way is just to put your camera on manual focus, take a picture and then change the focus a little bit, take another picture, change the focus. So you can do it that way. And that way you can take these focus stacked photos, even if you have a very inexpensive, very old, like DSLR camera. And then a third way is to move the camera. So you can take a picture and then shove the camera a little bit closer, another picture, move the camera closer, or use a focusing rail. So that's an attachment to your tripod where you turn a knob and it moves your camera a little bit closer every time you turn the knob. And then you back of pictures that way. But definitely the easiest and fastest way to do it is with one of the newer cameras. And recently, I've been using this Olympus camera, and it has a feature called focus stacking. And what it does is it takes all these pictures, up to 15 of them, and then it combines them all together in the camera. So I don't have to do anything on the computer. It just takes 15 pictures really quick, and then takes about 10 seconds to combine them into one final image. And that's how I've been taking pretty much all of my pictures for the past 6 months or so. Because if I use the Nikon, then I take all these focus stacks, and if I'm out in the forest for 6 hours, then I have 6 hours of computer work to assemble all of these stacks and get them all into good shape. Whereas if I use my Olympus in the focus stacking mode, I can just do that all automatically on just right on the camera. But one thing about the Olympus is that it saves all 16 images. And so I wrote this program which renames just the stacked ones. And so here are my photos that I took August 12th on Telluride. And if this program that I wrote is called Stack Copy. And so if I just, well, if I do a dry run, it'll show me all of the photos that are actually stacked. Take that off. And now it's renaming all of these photos. So I took 60 photos on August 12th, and it just renamed them. So the ones that are actually stacked have a different file name. Now, if I go back into Lightroom, let's see over in the Lightroom here, go to August 12th, then I resynchronize this directory, then only those stacked photos will come up. And that way you don't have to like sort through 16 pictures for every stack you took. It just only shows you all of the ones that are actually stacked. And that should come up in a second once it imports them, and then you can just really easily upload those to iNaturalist or whatever you do with them. And usually shoot them in such a way that there's no processing at all needed. Like, for example, here's one here. If we just look at that one, this is the photo directly as it came off my camera, nothing else needed. And you can see, it looks really good. There's like, I don't have to do anything with Photoshop or anything like that. These are just pictures. And so you can see I've been doing a bunch of flowers out because it was a little bit dry in Telluride. So I was getting some good flower photos and stuff like that. But when people ask me what camera to buy, usually I'll recommend the Olympus. It's not a good choice if you're doing bioluminescent or very low light photography, but the Olympus has a smaller sensor, and so it's a lighter camera. It's less expensive. The lenses are excellent and lighter than they are with other camera systems. So I usually tell people to get Olympus, but not the Olympus TG, because those are basically like cell phone photos. There's a bunch of different Olympus models, but one of the ones with interchangeable lenses, and then you can get world quality photos for not too much money. So another program I like to use, let's see, this one is to analyze DNA sequences. Uh, and this one is called Mega 12. And so Mega is this science program here. And what we'll do is build a new alignment, retrieve sequences from a file. And that should have some sequences of some stuff from Brazil. So I was in Brazil a couple years ago, and I found this psilocybe there. And it was collected and sent to Kew Gardens in the UK. And I just got the DNA sequence back from it a couple weeks ago. And turns out it is a new species. And so here is the sequence of that one right here. Um, and so what I can do, let's see, I will just delete most of the sequences out of here just so it doesn't take forever. But here we have 30 sequences, including my sequences. And if we look over to the alignment, you can see it's like a whole big jumbled mess. So the first thing we do is we go align by muscle. And this will vertically align all of these sequences up and down so you can directly compare each position. And then you can do phylogenetic analysis. So here you can see they're all neat. And this is kind of cool because like around here, this is a conserved region. All the sequences are the same. And then here's a variable region. So in this group, which is the Psilocybe zapotecorum group, this position here, or over here, these are where they really vary. So then if we want to really compare these, we can go to phylogenetic analysis. And it's not protein coding data. And then to make a really good tree, we'll test all the models. So there's a whole bunch of different mathematical models that we can use to try to reconstruct how these things evolved. And now it's testing a whole bunch of them. And it found that the very best one for this data set is K2 plus I. So we go over to phylogeny, make a maximum likelihood tree, use this data, and it's already set correctly. So this is our K2 plus I. So now when I build a tree, it'll make the best tree that we can do, and it's calculating exactly how these 31 sequences probably evolved. And so there's our tree right there. And so it's kind of cool because you can see exactly how your mushroom fits into the whole tree of life. And being in Colorado, you guys get free DNA sequencing at least through the end of the year. So you should be collecting all the mushrooms that you see, sending them in to Mycology lab in Michigan for DNA sequencing. Because just a couple of years ago, it was about $30 per collection to get your collection DNA sequenced. And now it's free for as many as you collect. So you can send in one, you can send in 100. This year, I sent in 2,700 bags of mushrooms to my garage, and I've been getting really good results from all of that. It's really neat just to see exactly what it is that I've been photographing and finding. And whole lot of them are probably undescribed species, and it really makes it easy to kind of dig into that when you have the DNA barcode. Let's see. Another thing that I like to do is identify mushrooms on iNaturalist. So let me go back over to a browser here. And yeah, go ahead. Um, Most mushroom species in Colorado have been sequenced before. So I've been processing a whole lot of Colorado sequence data, and usually there will be 100% matches with other collections. And, it doesn't always tell you what the mushroom is, but it does tell you which other observations are the same species. But yeah, most of them have a new one. So I'd say like maybe like 5% of them that we sequence from Colorado are novel. And then we're like, wow, we've never seen that one before. And it really depends on what you're collecting. Like if you're collecting little brown conocebes or something, then your chances of finding one that's never been sequenced before are very high. If it does have any matches, it's usually from Arizona. Whereas if you're getting something like Russula or Cortinarius, people have been doing sequencing a lot of those, so you'll probably get matches for those. But occasionally we can sequence something and then know exactly what it is because the holotype has been sequenced or some really good collections, and then we'll say, okay, it's definitely that. But more often we just put code names on things, and so we have a code name for all of the sequences that we don't know exactly what they are. And that way, at least we can keep track of like which species we have, and even if something's not been formally published, we can at least have a name to call it. So here I have this is the identify interface. And so I'll put in Colorado. And usually what I'll do is when I'm traveling wherever I am, I will try to identify all the mushrooms that are found in that state. Uh, Helps other people know what they have and get them excited about iNaturalist. And it also helps me know where the mushrooms are, what kind of shape they're in, maybe some place I would want to go out and see. This one here, Looks like it was uploaded 3 minutes ago, and they think it's Gloeophyllum. I agree. I think it is, but I'm not 100% sure. It's really difficult without the underside. Does anyone else have an opinion on which mushroom this is?
The dry one.
It's dry, but I do think it is Gloeophyllum sepiarium. But a lot of people here, they're not really mushroom experts, so they don't know that they need to show the underside. And so I find when I'm on here, I'm typing like it would really help to show the underside about 300 times a day. So I made a macro. All I do is hit F2, and then it says it would really help to see a close-up photo of the underside. Or I do a lot of identification in Mexico and South America, so I hit F3. It says the same thing in Spanish. But it's really fun just to flip through here and kind of help people identify stuff. Scutellaria scutellata requires microscopy, so I'm just going to bump this one down to Scutellaria. Oh, that's really cool. That's one of those truffles found by Heather Dawson. But when I was a Telluride, I was able to, I went through and identified all of the other, whoa, that's a cool Gymnopilus. That is really scaly. I think this one is Gymnopilus fulvosquamulosus. Pretty sure that's the only one that can get that scaly. That's a really cool one. I would love to find that cluster. I don't have any really awesome photos of that one yet. Or like Marasmius aureates. Yeah, that one definitely is. Let's see, one more program I will show you on here is called Pixie And Pixie is what I use to measure all of the things with the microscope. So I'll take a picture with the microscope. And when I'm at home, what I do is I just have a Nikon camera mounted to the third eye of my microscope on the trinocular port. And then I have a remote. So if I want to take a picture, I just press the button and it takes the picture. More often, what I do is I hold down on the button as I slowly turn the fine focus. So it ends up taking like 30 pictures, and then I load those into Helicon Focus and combine them. Because the more magnification you have, the less depth of field you have. So the focus stacking is really helpful with microscopic images to be able to get everything in focus at the same time. So these are some scleroderma spores. This is an undescribed scleroderma that I found in California. And so what I do is I open up Pixie I load this into Pixie and then I just click on the spores. And we'll include the ornamentation in this. So we'll just go from there to there and from here to here. And then you can see that, let's see, you can see that these are measuring the spores. It actually looks like it is measuring them wrong. Um, I guess, oh, I know why. It's because I use gigapixel to like expand this image, and then this program does not know how many pixels per micrometer it is. But over here you have all your calibrations, and so I tell it which objective I'm using and which camera, and then just click on the edges of the spores, and it goes ahead and measures everything. Um, and then the cool thing is that it gives you these really nice statistics over here. So here it shows what the smallest spore is, what the largest spore is, the Q value, the mean, the average Q value, all this stuff that you're supposed to calculate. But it takes a while to calculate if you don't have this program. So this is a free program. Unfortunately, it only works on Windows. Um, so on Macintosh, people use like ImageJ or a few other programs, but usually I'll measure 30 spores in here, and then you can just copy this onto the clipboard, paste it into your iNaturalist observation, and have a really good idea of exactly what the spores are, what size the spores are, and then you can do the cystidia and all the different microscopic features. And that is mostly all I have for you. So I'll take questions in a minute. but I'll mention just really quickly about modifications you can make to your car that are really useful for mushroom hunting. Um, One modification, I have the Subaru Legacy, so it's the Subaru that has a trunk that can close for security. But one thing I noticed is that if you smash the window and hit the trunk release, then the trunk just opens right up. And so I decided that since I have a lot of camera gear and I'm always traveling, I don't want it to be like that. So I took apart my dashboard and disconnected the trunk release. And that way there's no way to open the trunk without the remote. So the trunk is pretty secure, almost like a safe. And no one could just break the window and pop your trunk. So I always modify that. And other cars, I'll like disconnect that little cable that goes from that trunk release pull back to the trunk, and that way they can't pop your trunk that way. Another thing I like to do is for charging, a lot of times with vehicles, you have to turn on the ignition in order to charge your phone or your other devices. And when you have the ignition on, a lot of times you can't lock your car. And also it takes like a lot of electricity. Like when I turn my ignition on, it pulls 12 amps of power, even though charging my phone only pulls 1 amp. So wasting a lot of electricity in my battery by having the ignition on. And so what I do is I wire directly to the battery. First, it goes to a fuse for safety, and then I run the wire through the firewall in my car, and then wire up the outlets in the car, like those 12-volt outlets, so they go directly to the battery. And that way I can have my car closed and locked. It can just be charging overnight. You don't need the ignition on to charge your things or anything like that. And so that is a pretty useful modification. And another thing that I do is I put a 2000-watt sine wave inverter in my trunk. And that way, I just take like the same kind of cables that you would use to install a subwoofer in your car and have the sine wave inverter in my trunk. And then I can have 2000 watts of power, and it's really pure sine wave power. So I can run things like a printer. So I carry a laser printer so I can print out those nice iNaturalist labels, no matter where I am. And also you can run a PCR machine. And so you can bring a PCR machine and some DNA extraction stuff and primers and all of that. Just keep it on ice in your cooler, and then you can run PCR towards the end of the day. And then when the PCR finishes, you just drop it in the mail, and you'll have your DNA barcodes in a couple weeks or a couple days, even if you're on the road. The way I like to do this is, the PCR machine takes a lot of electricity. And so I will set up my PCRs, and then I will drive to the next location. So, PCR takes like 2.5 hours to run. So if I'm driving anyway, then, I'm not having to like idle my car for a couple hours while the PCR machine runs. And one last thing is that I like to install a voltmeter and an ammeter in my car. And so I just, for about 30 bucks on Amazon, you get a voltmeter and an ammeter thing. And what it does is it shows me what voltage my battery is at. So when I'm like out in the middle of nowhere, maybe using my laptop or the microscope, then I will just kind of glance up and see what the car battery is, and I'll know exactly when I have to start my car in order to not have to jump it. And then I carry one of those supercapacitor things. So if I do not pay attention and I have to jump my car, then I just grab that and jump it, and I don't have to like flag somebody down to jump the car. So those things are really good. And then it has an ammeter too. And the ammeter has a wire that goes to the battery and clips onto one of the battery terminals. So you can see exactly how much power is going into your battery and exactly how much power is coming out of your battery. And that's really neat because you can see like exactly how much power your laptop is taking, your microscope, your PCR machine, and you can like kind of know exactly how much you're draining your battery. And then when you run your car, you see how much power is going back into your battery, so you know how long you have to run your car before your battery is fully charged again. So it's really nice to have that, just to be able to keep an eye on the voltage of your car battery and everything. So I think that is just about everything. Does anybody have any questions about any of these things?
All pretty good, Alan. Oh, yeah.
Thank you.
They have a photo of the philosophy from Brazil.
Yeah, I got the photo of Salacity from Brazil. Um, yeah, they're actually pretty cool. Um, yeah, I can put the QR code. Let's see. QR code is over here. Probably wanna hide this thing.
What was the name of the power inverter, the 2001 power inverter?
The name of the power inverter, the brand doesn't really matter, but what does matter is the kind of waveform it generates. So if you're in your house, you get sine waves, a very nice smooth wave. But if you buy like a $40 power inverter at Walmart, it'll make square waves. And those are very noisy waves. And square waves usually work, but if you try to run a printer off of a square wave power source, for some reason it doesn't work. And it's really best to have sine waves. So just any 2000-watt sine wave, like it's actually, they call it like a pure sine or real sine wave inverter is the right kind. And you can get a really good one for about $120. So not super expensive, but yeah, definitely worth it. And it's also nice because you can just put like a soldering iron or whatever, and I can like make all my modifications to my car and solder everything, just, do a really good job, even though if I'm in the middle of nowhere. And that's psilocybe from Brazil. Let's see. I will try just load up this presentation. I think I added it to here. So this is one of the talks that I gave in Telluride. It is new discoveries in the taxonomy of psilocybin mushrooms. And let's see. Well, first I'll show you this one. This, okay. This is the psilocybe that was found in Telluride. And so this is a photograph by John Plischke. You can see it is staining blue. It's got a ring on the stem. So I think that makes it Psilocybe mescaleroensis, which is the one that's described from the Sacramento Mountains in New Mexico. This is the one where Brian Barzee and I went out there in 2016, found the guy who discovered these and ended up getting good photos of them. That was definitely an epic trip. So I think this is the same species, but this was found in the Telluride area. Unfortunately, he did not save this sample. So somebody probably has it, but nobody really knows which one this is. Maybe it's a new species. Definitely have to recollect it on a good year. Also, Paul Stamets in his presentation, he had a photo of a mushroom that was definitely psilocybe that was collected in Telluride. And that one is had a much longer stem, definitely not the same species as this. So there's at least two psilocybes in Telluride. One of them is probably Psilocybe aztecorum, which is really common up in the above the Netherland area. But to answer Brian's question, this is the new species of Psilocybe that I was finding in Brazil. And so it has the same odor and taste as Psilocybe zapotecorum. So it tastes like cucumber and radish. And like Malircula, the gills stay really white until the spores mature, and it really likes these landslide areas. So I found it like in the muddy creek banks next to a creek. And it's similar to Zapotecorum, but much more yellow on the cap. Like Zapotecorum is never like canary yellow like this. There it is on black velvet. And yeah, I was really happy that I was in Brazil with this amazing mycologist named Larissa, and she had a permit to collect there. And so she was able to collect all of these. And now they're in the herbarium at Kew Gardens in the UK. And she was able to do DNA sequencing on all of the collections. And yeah, so it's a really cool thing. And the barcode matches 95%. Well, is the closest match to anything, and that's pretty far, especially in this section. So it's definitely one that's never been sequenced before. We'll have to do a bunch of microscope work to see if maybe it has been described but has not been sequenced. There's a few names that it could be from South America, but definitely a pretty cool species. Yep.
Had a permit.
So in some places you can just pick mushrooms and it's totally legal. In other places against picking mushrooms and they only give permits. to certain people. And so in South America, a lot of times they're kind of locked down. So if you're not on private property, you would need permission from the government to collect mushrooms. And so she had this whole research project and did all the paperwork to get government permission to collect mushrooms. And so we were able to legally collect everything and then put it all into the herbarium.
Yeah.
Yep.
How is the DNA sequencing done? Like, do they have
How
to-
is DNA sequencing done? That is a really good question. And let's see, I actually wrote a web page that shows you that. So it's just this wiki counterculture labs DNA sequencing. And so this has a whole bunch of videos and links, but basically DNA extractions. So you just get the mushroom into solution. So you take 1 milligram of mushroom, it's the amount that you can barely see, put it into a PCR tube, put this DNA extraction solution that you can make at home in there, and then heat it up for 10 minutes, and then you have your DNA in solution. And then you run PCR. So PCR stands for polymerase chain reaction. And that's the same chemical reaction they were using to test for COVID, but it's much more interesting just to put mushrooms in there. And what it does is it copies just certain parts of the genome. So you put in the mushroom extract, you put in a forward primer, a reverse primer, and then the master mix. And the forward primer tells it where to start copying, the reverse primer tells it where to stop copying, and it makes billions of copies of whatever gene you want. And then you can do gel electrophoresis to test if the PCR worked or not, or you could just proceed to sequencing. And Sanger sequencing is the kind that most people do at home because it costs $5 each. And you can just send a few of them off to this lab. Or like what I do is if I get up at 9 in the morning, start the PCR at 9 a.m., it'll finish by noon, and then I fill out the order form. The guy stops by my house at 2 p.m., and then by midnight, I have the results in my email. Because the sequencer itself is kind of expensive and it doesn't really make sense to have one at home. But now they have nanopore sequencers. So if you're doing hundreds or thousands of samples, you can sequence thousands of samples for about 30 cents each. And so a lot of people are doing that, but you need a lot of samples to do that. But in any case, it's pretty easy, like to have a DNA lab in your house. It's under $1,000. You just need the PCR machine and some pipettes and some chemicals. But since you live in Colorado, you don't have to mess with any of this. You just dry the mushroom, write the iNaturalist number on the bag, and drop it in the mail. And then a couple months later, the sequence will appear on your iNaturalist observation.
Do you have the address that they?
Oh, that's a good question. So you just go to mycoblitz.org, and it's got all sorts of details about that. Is mycoblitz.org even up? Maybe my internet is being funny. So here's the mycoblitz.org. You can see that Summer Continental Mycoblitz is August 8th through the 17th. And but since you're in Colorado, you can just send stuff collected anytime. And let's see. Here it says you're supposed to print out voucher slips, but really it's fine like to write the iNaturalist number. That's the most important part. And if we scroll down, we can probably get the address here. There's a whole bunch of questions. Yep, here it is. There's the address to send your specimens into.
What gives Colorado?
What gives Colorado the privilege to have free access to DNA sequencing? That is a really good question. And let's see if I can.
And your specimen. Where's the rule? Where's the rule?
I think it was maybe a little bit more complicated than that, but I think somebody in Colorado maybe annoyed Stephen at one point, and Stephen decided to just get a ton of sequences from Colorado to kind of leave that person in the dust. But that was before my time. I don't know anything about that. In any case, somewhere, let's see, somewhere on here, there's a whole map, and there's about 10 or 15 states that have open call, so they can just get free DNA sequencing in that state. Let's see, you got a question back there? That's a good question. How do you access GPS coordinates for an observation on iNaturalist? So if I go just to my observations over here, we'll just go to the first one. This is a bear. I did not get a good picture of the bear, but it was definitely a bear. So you go over here and click details. And then here's the GPS coordinates right here. And then the accuracy is important to look at. So the accuracy is 3 meters. So some of the observations will have an accuracy of like 2 kilometers. But you can see, when you zoom in, you can see all the other bears. You can see bears are not uncommon around this part of Telluride. So usually I'll just like click on this to copy it and then go Google Maps and paste it in there. Oops. Paste it into Google Maps, and then you can have it direct you right there. So this is pretty nice. Like if I'm identifying mushrooms on iNaturalist, yeah, you can see that's exactly where I saw the bear right there. If I'm identifying mushrooms on there and somebody finds something really rare, then I can just like go to that exact spot and get good photos of it. Let's see, yep, that. Thoughts or recommendations on microscopes? Yeah, so I usually recommend people get an Amscope B120 because this is the least expensive microscope that works really well for mycology. And wow, the prices have really gone up since I looked at this last. That's the Trump tariffs. But yeah, up to about $300 for the Amscope B120. And so this microscope is a compound scope. It's actually quite good. It's not a toy. It's got the features that I look for in a microscope are the mechanical stage right there, the 100 times oil immersion objective. So that means that you can magnify things 1,000 times, and the binocular eyepieces. You have these two eyepieces. Here it says 40 times to 2,500 times. But that's because they give you 10 times eyepieces and 25 times eyepieces. So what you should do is when you order one of these things, as soon as it comes, take those 25 times eyepieces, throw them in the garbage because they're empty magnification, and they just have that so they can advertise 2,500 times, but it's really 1,000 times. And those the 10x eyepieces are way better than the 25s. But you can actually do a surprising amount of work on this. Like really anything scientifically you can do. The only reason you would spend more money than this is to get better image quality. So if you spend like $5,000, you can get crystal clear images, although these are pretty clear. It's kind of like camera lenses where the cheapest lens is clear, but then a little more expensive lens more clear, but then like a $20,000 lens is just a tiny bit more clear than that. Um, so there's, you can like really go down the rabbit hole, but I think people should get this one because it's really cheap and it's really good. And then if you find yourself through it all day, every day, and you actually like it, then you can start to upgrade it and get a more expensive microscope. But most microscopes just gather dust. And the reason for that is microscopy is extremely difficult. It took me a lot of years to learn how to do it well, and it's also extremely time consuming. So, the DNA barcoding and just like throw it in the mail, super easy and fast. But to do a really good job of microscopy on just one collection, it can take me all day. And that's like 4 hours actually doing the microscope work and then 4 hours working on the photos on the computer to assemble them and measure them and write the description and everything. So, just to look at the spores is like 5 seconds of work, and sometimes that's all you need, just like one look at the spores, you know exactly what it is. But other times, like if it's a gymnopilus or something, you have to do a full microscopic workup. And it's really fun. It's really cool to see that whole other layer of beauty that are in mushrooms, but it takes a special personality to really get into it. So Most microscopes are just gathering dust. And I can't really see you raise your hand. You guys raise your hands because of the lights in my face. So just if you have questions, just shout them out.
So in your bag, you have a 30 micro lens and 90.
Yeah.
Why those two only? And is there a zoom one?
Yeah, that's a good question. So in my bag, I carry a 30mm macro lens and a 90mm macro lens. Why those two lenses? And is there a zoom one? And the answer is no, there is no zoom one. These are both macro lenses, which just means that you can put it very, very close to the mushroom and see really good detail. But macro lenses don't tend to be zoom lenses. The 30mm lens is razor sharp, but the working distance is only like an inch or so. So you have to get it right up close to the mushroom, which is fine, except that it can be hard to get light in there. And so it's also very tiny and very, very lightweight. So, it's a nice lens to have, but I usually use the 90mm lens. The 90mm lens is a much narrower field of view. So 30mm lens, you can kind of see the whole forest. 90mm lens, you're really only seeing the mushroom. It's much heavier and oh, okay, yeah. The 90mm lens is much heavier, but it has a much longer working distance. So like if you're taking a picture of like a dragonfly or something, if you try to get up there with a 30mm lens, you're just going to scare it away. Whereas the 90mm lens, you can get a good picture of it without having to get too close with it. And there's also like a 60, but a 60 is sort of redundant when you have those. So I wanted to get the macro lenses that were the most different from each other. And that's why I chose the 30 and the 90. Yep. That's a good question. What are the steps involved for discovering a new species? And the best thing to do is just to Google how to describe a new fungal species. And then this excellent paper comes up. Let's see, looks like I've typed that again. So this paper here comes up, and this is the first author here is Kathy Aim, who's a really amazing mycologist who was at Telluride, and she gave a talk on how to describe new species, which was really good. But in this paper, it goes and tells you exactly how, here's all the nomenclatural requirements and how to make a good type collection and best practices for taxonomy, all that stuff. So if you follow these guidelines, then you can publish a really good paper on a new species. And it doesn't really cost any money to describe a new species. There's a lot of free journals you can submit it to. They don't check your identification or credentials. Anybody can do it. And so definitely something that really needs to be done because there are a lot of undescribed species out there and we need names for them so we can communicate with people as to what exactly we found. So yeah, that's a really good thing to do is like if you're out foraging, just start making collections. And when you send them off for DNA barcoding, since they only use a milligram for DNA barcoding, you can just send them like a little piece, like maybe the size of a dime, and then keep the rest of the collection at home. And that way, if it turns out to be a new species, then you have the collection there, and you can bring it over to Andy at the Denver Herbarium. Give him that for a holotype. It's a good idea to study more than one collection, so study a few collections, though it's not a requirement. It's a good idea to make a good collection, like several fruit bodies at different stages of development, though again, that's not a requirement. Um, and then you got to make a Latinized name. So there's still there's a lot of leeway you have in like what you can name it. But if you name it like after your friend in 10,000 years, the name won't really mean anything. So I think it's best to name the species after like a unique feature that separates it from its closest relative. Um, but yeah, this paper is definitely a really good place to start. Yep. Yeah, my path into mycology. So, I was born in Chicago and grew up in the Chicago suburbs. And then when I was 15 years old, I got expelled from high school for sending thousands of pages of faxes to the school fax machine from a payphone. And so I had a lot of time off, and I spent my time off getting really good at breaking into computers. So by the time I went back to school, I was like hacking army bases and big corporations and things like that. And I've always been really into nature, doing a lot of camping. My parents are really into nature and stuff like that. And so I started to get into mushrooms like in 2001 when I started to see a lot of mushrooms around. And I just started taking pictures of them, started saving all of the mushrooms. And I decided that I wanted to be a professional mycologist. And so I decided that the way that I would do this is I would be very helpful on social media. So whenever anybody posted a picture of a mushroom or had some kind of mushroom question, I would answer it. And that way, when somebody needed to hire a mycologist, they would be like, "Oh, I know a guy. Alan knows a lot about mushrooms." And so that worked out really well. Now I'm kind of traveling the country, going to different mushroom events every weekend. So I've been to different events kind of every weekend for a long time, and I have a whole bunch more scheduled. And it's been really cool because I get to teach a lot of people a lot of things about mushrooms and photography, and then lead lots of trips all over the world to look for mushrooms. And I also decided that, the DNA barcoding was a really cool thing to do. So I just started doing that at home. And the whole strategy of posting on social media worked out pretty well because in 2018, these people asked me if I wanted to work for their mushroom company, and I ended up being the chief mycologist for their company. And it was one of these jobs that paid well, and I could just do whatever I want all the time. And that lasted for a few years, but then that company ran out of money. So now I just travel around, look for mushrooms. But basically, I just taught myself everything that I know about mushrooms and just spend a lot of time in the field looking at mushrooms and a lot of time in the laboratory studying the ones that I found. Mhm. Yep.
Hacking Kali or a black box or
Yeah, I guess the question is, what kind of tools do I use for hacking? And I do have Kali Linux installed on let's see, I think it's my other laptop, but I don't really use Kali Linux because I'd rather just install the tools that I have. You can just install the tools on any Linux. So this particular one I'm using right now is WSL, so it's Windows Subsystem for Linux. It's really easy because if you just have Windows running, you can just go ahead and install Linux on it. And it's like, real Linux. It actually works just like Linux. I use, pretty much anything in here that you could do with Linux, you can do under here. I mean, it is actually Linux. I also have VMware running, so I have like a few different distributions running in VMware, but typically I'll just use this. And um, let me actually show you one program that I made. This is kind of cool. This one. This is a program where what I did is I downloaded all of the iNaturalist observations in a CSV file. So it was all the research grade ones. It was like 113 million records, loaded them into Duck database. And now I can search if I just put like, I don't know, Rusala. It'll show me where Rusala, and I can put in any GPS coordinates in the world here, and it shows me when Rusala season is. Or I can put a Garicales, and it'll show me um, when just general mushroom season is. So this is a graph of when mushroom season is within 50 kilometers of my house. And so you can see in the winter, the mushroom season is very good. And then right now it's very dry, which is why I'm here. Um, But yeah, I don't really use any like special hacking OSs. I just like the tools I need and just go ahead and use them. Mhm. But I still do a bunch of like computer security and hacking type stuff. So if any of you guys like ever get hacked or need security advice, just let me know and I'll help you out and let you know exactly what you need to do to secure your devices and then be able to operate securely without hackers like messing up all your stuff.
You should come back next month. Tell us more about it.
I would be happy to give a presentation on hacking. Usually when I give my hacking presentations, it's like to college campuses and like computer security classes and stuff. But yeah, that stuff's really interesting. And, for me, it's really easy. I kind of grew up with computers. I think I got my first computer when I was 3 years old, like in 1980, because my dad was a computer science teacher. And so I can't even remember not having a computer, and I've always been really comfortable with them. So, it makes it, all this stuff, like it's all seems very easy to me because I was kind of born doing it. Yep. That's a good question. When I'm doing photo stacking, how are the pictures different? And the answer is they should be all the same except for where the focus is. So each picture is a small slice of the final image. And so I focus the camera really close, take a picture, focus it a little bit further, take a picture, focus it a little bit further, take a picture until I've gone all through the subject. So ideally, they're completely identical except for which part of it is in focus. But in practice, sometimes the camera's moving or sometimes they're not even using a tripod. So you're like, it's a little bit different, but it tries to compensate for that. And first it aligns all of the images and then it combines them in the photo stacking. So you can actually get pretty good results, even if you're just holding the camera in your hand or just using a cell phone. Like you can just use a cell phone and like tap on one mushroom, take a picture, tap on the one mushroom in the back, take a picture, and just do that 3 or 4 times, and then load those in and photo stack those. Mandy, Ecuador event. Yeah, that's a good idea. So we're leading a really cool trip to Ecuador, and it'll be like for 2 weeks in November, I think like November 7th through the 22nd, something like that. And we're partnering with Alex Bentley, who's a really cool scientist who lives in Ecuador, and it'll be in one of the most most biodiverse regions in the whole world. And we're going to 4 different places and 4 different elevation um, gradients there, and we see a ton of really cool mushrooms and plants. Alex is a reptile expert. And so we kind of have like experts on every kind of nature. We're bringing Joey, who runs the Crime Pays But Botany Doesn't YouTube channel, to identify all of the plants for us. So that'll be a really cool trip. And if you visit our website at mycena.llc, then you can, but it will be up on this website here, mycena.llc. Yep.
How selective are you which fungi?
How selective am I for which fungi I document on iNaturalist? Usually, well, if you like don't focus on anything, if you just like focus on all fungi, then you'll never get anywhere. But that's what I do. Anytime I see any mushroom, as long as it's in good shape, I will take a picture of it. But what I usually look for are very pretty fungi or very good examples of fungi. So I don't care if it's something super common. It might be like Cornacity apala that's in everybody's yard. If there's a whole bunch of them and they're in great shape, then I'll sit down with the camera and take a really good picture of it. But I also like to get really good pictures of the rare mushrooms. So if it's a super rare mushroom, even if it's not in good shape, I'll still take a picture of it. But yeah, typically I document absolutely everything. And then I save dried samples of everything that I photograph and get them all DNA barcoded because you don't really know what's cool and what's not until you get the DNA barcode data. And once I've DNA barcoded something a few times, then I'm like, oh yeah, it's just another one of those. But until you know what the DNA barcode is going to be like for the fungus that you see, it's a good idea to collect it and send it in. And most of these discoveries of new species and stuff are just people that didn't really know what they're seeing. They just grab every mushroom and document them all. And that's really valuable data. But if you do want to get anywhere in mycology, then you got to pick something and stick to it. Around here, I think like a really good one would be like Cornacida and Foliotina, because the little brown mushrooms, there's a ton of them and they're very poorly known. Or you could do Melanoleuca, Rusula, Cortinaria, any mushrooms that you like. You don't have to only collect that, but just pick any genus that you think is cool, and just every time you see it, pay special attention to it and collect it. And then in just a few years, you can be the world expert on any genus of mushrooms. All right, well, thank you very much. Thanks, Greg.
So our next meeting's at Cook and Taste, and we are September 8th. Here, but at Mitchell Hall, it's not in this venue, it's in Mitchell Hall. So, thanks for coming.